Okay. Destiny, can you hear me?
Yeah, I can hear you.
All right. I think I got all my issues fixed. So we can go forward from here.
Okay, cool.
I'm just going to go ahead and start from where I left off in the last lecture, which is on slide 14, which is translation. So let me go ahead and share this PowerPoint. All right. And I hope that you can see this. I think you can.
Yeah, I can see it.
Okay. So we covered transcription. So we're going back to just to remind you and everyone else that we're talking about the central dogma, which this term was coined by Watson and Crick. I don't even think they really knew what it meant. So, but it stuck. But really, it's kind of referring as the central idea of how the information that's stored in DNA is converted into proteins, and then specifically through that intermediate
Yelling out again.
RNA. So DNA is there's only one copy, and it doesn't matter if you're a prokaryote or eukaryote. And that single copy of DNA is either stored in the nucleus or in the cytoplasm, and that has to be converted into RNA. No matter what organism you are, except for viruses. Viruses are the exception. But all other things that meet all the definitions of having a cell have DNA that's converted into RNA. When that's done, we call that messenger RNA. So I'm just going to put an M here because we learned in chapter 5 that there's lots of different kinds of RNA. There's tRNA, and we're going to talk about that now, but there's transfer RNA, and we have ribosomal RNA, and that's what the T and the R stand for. And this stands for the messenger. So this is the one that the DNA is made into RNA. And this process is called transcription. And we just covered that in the previous lecture. So the next step, once the mRNA is made, it goes out into the cytoplasm. Or if you're a bacteria, it's already in the cytoplasm. And then it's read by ribosomes, which we talked about in chapter 6. And ribosomes are basically free or bound that we talked about. You know, the bound ones are on the rough ER, and the free ones are in the cytoplasm. So regardless, the ribosomes, it doesn't matter what message it is, and this is kind of how viruses hijack your system. So viruses can put in messenger RNA, and in fact, that Pfizer vaccine, the COVID vaccine, is actually artificial mRNA that's introduced into your cell, and it uses your own ribosomes to make the proteins for that spike protein that the coronavirus uses to get into the cell, and it makes antibodies. So basically, the COVID vaccine is putting mRNA into your cells, and that's getting turned into proteins by your own cells to make the spike protein, which it recognizes as foreign, and antibodies produce this. So it's basically just skipping this transcription step. Whereas the other virus, the Johnson & Johnson virus, it just directly puts the proteins into your bloodstream. So it's not going through this step. All right, so anyway, the next step is to convert the RNA into proteins by ribosomes, and that's called translation. And we briefly talked about this previously, but we're going to kind of get into in-depth in this lecture. So we talked about that the RNA is has 4 letters in its alphabet, like English has 26, and proteins have 20 letters. And so if we were just relying on RNA, we can only make 4 different, we can only have 4 different amino acids. So these are 20 amino acids. We have, if we assign, like, let's just say we assign tryptophan and phenylalanine and glycine and serine, we couldn't code for any other. Let's just say this would be A, and this would be G, and this would be C, and this would be U. We couldn't code for any other amino acids, so we would be missing the 16 amino acids from RNA. We would never be able to make a protein with any other except 4 amino acids. And so if we use 2 letters instead of 1, then we could code, that would be 2 to the first one is 2 to the second, which would be 4 letters. Or 4 possible outcomes. Sorry. 4 to the first, because there's 4 possibilities. Ignore that. 4 to the second would be 4 times 4, which would be 16 possibilities. But we still have 20, so that's not enough. So we actually need 3 letters, and that's 4 to the third, which is 4 times 4 times 4, which is 64. So we have 64 possible combinations if we use a 3-letter code. And so that's what ribosomes have evolved to read the 3-letter code. And we call that 3-letter code a codon. So here's the DNA strand. This is the artist example, and you know that DNA is double-stranded. So we talked about genes and parts of the genes previously. So we're just gonna zoom in on a gene, and you know up here there's a promoter, and down here there's a terminator, and this is the coding region. It's just a short one that the artist is using. So we know that the mRNA can only be read or made from 5 to 3 because it needs that hydroxyl group to do the dehydration reaction, which we learned about in chapter 5. Okay, so it turns out that if the DNA code is 3 to 5, then this mRNA has to be 5 to 3. So it can only read one of the 2 strands, and then there'd be another strand. And you guys did practice problems on this in the last lab. So in transcription, the RNA polymerase would see an A and it would add a U. Right? It would see a C and then it would add a G, and then it would see a C and add a G, and so on. And then this would be modified with the 5' cap and the poly A tail. And then when the ribosome would find the cap, it would find the G, and it would come in and it would translate this. So it would look for this 3-letter code. So this 3-letter code here from the DNA sequence ACC would be UGG. And UGG, when the ribosome sees UGG, it will insert, using a transfer RNA, and we're going to talk about that in a second, but it will insert the amino acid tryptophan. And you probably don't remember this, but remember we have an amino and a carboxyl terminus, and we always go from the amino to the carboxyl. And the reason is because we need that hydroxyl group to make water to do the dehydration reaction. So that's why amino acids are always made from the amino to the carboxyl end. So the next 3 letters in the DNA would be AAA. That would be read by the RNA polymerase as UUU during transcription. And then in translation, the ribosome would read that as phenylalanine, and so on and so forth. And so you guys got a lot of practice doing this when you did the lab. Well, hopefully you did the lab. And if you haven't done the lab, you might want to do the lab to practice these things, because you will see this sort of stuff on the exam. All right, so make sure you know the term codon. Make sure that you know that DNA is, the DNA template strand is going to be 3 to 5. During transcription, the mRNA makes it from 5 to 3, and that the 3 letters are going to be the codons. So this is kind of like a fake one, because every protein starts with methionine, but I might ask you something like this on the test just to simplify it. And then I might give you a DNA sequence like, well, I'll just give you the example on the next slide because it'll be easier. So this is the genetic code. Scientists figured this out by just putting fake mRNA, just kind of like the COVID vaccine, the Pfizer vaccine. And so they just put a long strands of A's in there, and then they wanted to know what proteins came out of it. So then they examined the proteins. And so if there was long strands of AAA, which is shown here, then the protein would be lysine, lysine, lysine. And you guys don't need to know or memorize this. On the test, I will give you the genetic code if you need to use it. So you don't need to memorize this. That's like memorizing the periodic table. But this AAA codes for lysine. And if there's a, so let's just do an example. I'll just do a quick example. Remember, the DNA template has to be 3 to 5. So and remember, DNA has T's, not U's. So I'm just going to make up a DNA sequence like I would for a test. And, you know, we're ignoring methionine as the start codon, but in real life, it would always be methionine. And so this is what I would give you on the test. I would say, here's a DNA sequence. What is the RNA that's made out of it? And so you should be able to do this. A pairs with T, but on RNA it's U. T pairs with A, and then C pairs with G. And we can just go down the line and do this. And just make sure that you guys know the pairings. And don't forget the 5 and the 3' ends, because that's really important. And then, so the ribosome would come along and it would read this, and it would read it in 3-letter batches. So the first 3 letters would be UAG. And so I would give you this code, and you could go look. You could look for the first base is U, second base is A, and then the third base is G. So UAG, and that's a terrible example. This would be a stop codon, so it would stop translation. So let me make this a G instead. I should have known that. So I'll turn this into a C. So in this case, it would be UAC, and that would code for threonine. So this would be threonine, and then so the ribosome would move over 3 spots, and it would read CGA. And so we could look at this C and G and A. And so if we look, CGA is right here, and that codes for arginine, which would be the next one. And then remember, it's always amino. And then the next one would be UGA. So we could go UGA, and that just happens to be a stop codon. So you don't have to put stop in there. The ribosome would just stop on its own, and then you would be left with a carboxyl end. So this would be the 2 amino acids that would be made from this particular DNA sequence. And that's kind of the stuff I would ask you to do on the test, giving you this table. So like I said, the lab that you guys just did, I just want to make sure you get the name of the lab right. So the gene expression lab is the lab where you would be able to practice these problems here. Okay, so the reading frame is really important. In this case, there's 3 possible reading frames. So one of the reading frames would be what we did. If the ribosome jumped over, that's not a very good color. If the ribosome jumped over 1 spot, it would read something different. So instead of reading ATG, would read TGG, and that would make something completely different. So the TGG, this would be made into the codon ACC, and ACC would be here. So that would be threonine. So it just happened to not change it. And in this case, the next one would be CTA. So we would get GAU. So GAU would be here. And that's aspartic acid. So and then we would have another reading frame here. So this is a different, the point that I'm trying to make here is that if the reading frame, if the ribosome accidentally moves over one letter, it's going to make the wrong protein, which is going to fold into the wrong shape, and it won't have the right function. So the reading frame is really important. And then the final reading frame would be if it moved over one, instead of one letter, it moved over two. And so it would read like this. So this would be the third reading frame, which would be CCG. And we could look and see what CCG is. And CCG would be proline. So this would be proline. And then the next one would be TAC, which would be read as UAG. And so, I'm sorry, AUG, which is methionine. And then it would end. So all three of these would make a different amino acid, depending on what the reading frame was. So the example that I like to use is, it's kind of like if we had a three-letter sentence. And so let's just say the sentence is the fat red cat. So if we moved over one letter, then this is what the sentence would read. T, have, utter, at. That doesn't make any sense, right? And it's the same thing with the cells. If it moves over one letter, it's no longer the same sentence. It means something completely different. And again, if we moved over two letters, right, then that would be a bunch of gibberish as well. If we moved over three letters, it would put us back into the reading frame. So we would still read part of the sentence. We would just lose the THE part, which wouldn't affect the protein as much as this. This is just totally destroys the proteins. So any three-letter shifts of the reading frame are not so bad. But any one or two-letter shifts are really bad. That would really wreck the protein, just like it wrecked this sentence. And then this is just, again, showing you what I just showed you. These three letters here, oops. These three letters here are, if we read them this three-letter code here and here and here and here, we would get a completely different protein by just shifting over one letter, because the amino acid sequence would be different, and that would be bad. That would make a completely different protein. You know, it might be a toxic protein. It could kill you. It could affect you in a bad way, and that's why, you know, if we go back and we look at, say, a mutation in the DNA, like let's say with this C accidentally got deleted because you went to the dentist and got some x-rays, that would change the reading frame of your protein. And if it was in something that was important, like involved in glycolysis, that would kill one of your cells, right? So it would be very bad if you were just a few cells, right? But since you're an adult and you have trillions of cells, it's not that bad. But you know, that's why it's bad to like be exposed to radiation when you're pregnant, or have, you know, young kids exposed to radiation, because they don't have as many cells as you to you know, just go, oh well, I don't care if I lose a couple million cells because I have a trillion. All right. So the way translation works is, I'm going to switch back to red. So this brown thing here is a ribosome, and ribosomes have 2 subunits. If you guys forgot that, you can go back to chapter 6 and take a look at it. So it has a large subunit, which is shown here as the big one, and then it has a small subunit, which is shown here as the small one. This is representative of the messenger RNA. Remember, it's always read in the 5' to 3' direction. And the ribosome reads it. It always makes, so this end would always be the amino end. And this end would always have a carboxylic acid, carboxyl end on it, because we need to do the dehydration reaction. So these things right here, the green kind of dynamite looking sticks, these are called transfer RNAs. They don't really look like this. This is an artist rendition. And these are the amino acids that they're going to pick up that are sort of floating around the cell. So the so there's an enzyme that links the amino acid to the tRNA. And it's called amino acyl tRNA synthase. And I'll show you that in the next slide. So that enzyme comes in, it links this amino acid to the tRNA. And once it does that, it's called charged. So charged means that it's a tRNA plus an amino acid. And now it's ready to go to the ribosome. So every tRNA has a 3-letter, what we call an anticodon. And that anticodon matches perfectly to the codon. So here is a codon, right? And so this is a tRNA. So this is UGG. So the anticodon here would be ACC, because A pairs with U, C pairs with G, and so on. Here's another codon. This one's UUU. So the anticodon would be AAA. Again, here's a 3-letter codon, and that's GGC. So the tRNA that would pair there would be CCG. And that particular tRNA would have a glycine attached to it. And this particular UUU codon would have a phenylalanine attached to it. And I like to double check these books because they have artists draw it. They're not really scientists, just to make sure that they did it right. We can go back to the genetic code table and take a look and see what UUU is. And we can see right here that UUU does code for phenylalanine. And so if the artist drew it right, then they would have phenylalanine here. And that's correct. UUU does code for phenylalanine. And we can check this one. GGC should code for glycine. And we can go back and look. And sure enough, if we go to GGG, sorry, what was it? GCC? GGC, it's also glycine. GGC, right here. Codes for glycine. So the artist did this correctly, and that's what we would expect. And in fact, we could go back. This one would be the one that would be right before, so that would come from this tRNA. And so that would be UGG, and so that should be tryptophan. So let's see if UGG is indeed tryptophan. And so we could go UGG is indeed tryptophan. All right. So the charged tRNA is going to go match with this anticodon. Once it binds in there, then there's going to be a dehydration reaction. Well, a hydrolysis to cut this bond from the tRNA, and then a dehydration reaction to link these two together. And then what happens is the ribosome moves over one space, and so this one would end up here. And since it's cleaved, it would have no amino acid on it. This one now has the growing protein chain on it, and it's going to end up in this spot. And then the next spot would be these 3 letters right here. That would be open. So it's kind of hard to visualize this, but you got to imagine that these brown ribosome parts are going to jump over 3 letters, and when it does that, where are all those tRNAs going to end up? This is kind of so this is the amino acid tRNA synthase. You should know this, so just write down a flashcard and remember it. I just remember that it's amino acid, so it's going to take amino acids and add them to a tRNA, and synthesizing is creating. So it's going to create an amino acid linked to a tRNA. And this is what it looks like. The amino acid goes into this site. The uncharged tRNA goes into this site. They get linked together. This reaction is dehydration. And it's done very specifically. So it has to attach to the 3' end of the tRNA, because that is the one with the hydroxyl group on it. So that it doesn't attach to the 5' end because it can't do the dehydration reaction to link it together. I don't care that you know this. This is the traditional way that scientists write this out because it's completely flat and it's easy to do. And you can remember DNA and RNA are like shoelaces. They're not like pieces of steel. They can move around. And so this RNA can make funky shapes by having sequences that bind to other parts of it. So this is kind of what we call a clover leaf representation. This is what the actual tRNA looks like in a 3D model. And then this is how the artist drew it, which is kind of what we're going to stick to. If you guys are taking a genetics class or whatever, this might be more important because there's a thing called wobble, and different tRNAs can code for the same amino acids, but you don't need to know that. What you should take out of this is that there's amino acid tRNA synthase. Every transfer RNA has a different anticodon, which gives it a different shape. So that means there's going to be 20 different amino acid tRNA synthases for every one of the anticodon tRNAs. And they can't be reused for different ones. Any questions so far? All right. So this is just we're looking at the ribosome here. We have the large subunit and we have the small subunit, and the mRNA goes in between, which is shown here. The sites are called E, P, and A, the 3 sites that we were just talking about, which are here. So this site is the E site, and that means exit. P is the peptide site. That's because it's the growing polypeptide. Remember, that's a fancy name for protein. The growing polypeptide grows from the P site. And then this is the A, this is the amino acid site, because the charged tRNA comes into it. So, a lot of students remember it as like Environmental Protection Agency, and I really don't care how you remember it. You should know what these sites are and what they do. So this is an artist drawing of it. Remember, it's going to read it from 5 to 3. And so on the exit site, we have a tRNA that's uncharged, and it's going to exit. It's going to go and get a new amino acid put on it. The P site is where the growing peptide is found. And remember that it's always on the amino end to the carboxyl because it can't do the dehydration reaction without that. And then the next amino acid will come in, and then this will get linked to this. So the A site is where the charged tRNA comes in. This is a charged tRNA because it has an amino acid attached to it. The E site is where the uncharged, or the one that doesn't have an amino acid on it, leaves at the exit site. and then the P site is where the growing chain is formed. All right, so just like in transcription and translation, we have initiation and elongation and termination. So in initiation, and I'm not going to get too involved in this, but remember we have that G cap, the 5' cap, and then we have the coding region. So let's just say it's AAAUUC, AGUA. And then we would have the poly A tail that's added onto that. And so in eukaryotes, the ribosome finds this G cap and it starts scanning. And it'll scan down until it finds the start codon AUG. So I'm going to make this a G. Sorry. So the ribosome will scan down until it finds this codon. And then once it does, the small subunit will scan. Once it does, the charged tRNA, it's always methionine. Methionine is always coded for AUG. And I'll just go back and show you that that's true. Wrong way. So AUG is always methionine. So every protein starts with methionine, and a lot of you guys might see in your comment section, you didn't start your protein with methionine. Remember, that's the start. So every protein that's made by every eukaryotic living cell, every protein starts with methionine because that's the start codon. That tells it where to start. All right. So it would start here. It would bring in the large subunit, and that would be so remember, this is the P site. So that would be the start. This is initiation. I don't care that you know this, but remember, A's are very closely chemically related to G's. Where we have you learned this in chapter 5. We have the purines and the pyrimidines. So these are purines. And so A's and G's are really closely related. So that means that instead of ATP, a lot of systems can use GTP as well. And that's what this uses to move the ribosome along. So that's what's driving this ribosome to pick up and move, 3 spaces towards the 3' end. All right. So from this slide, you should know that initiation, the ribosome finds the 5' cap, scans until it finds the first start codon. You should know that the first start codon is always AUG, even though I'm going to give that to you when I give you the genetic code. But this is a good thing to know. It's like knowing, the grammar rules, I before E, except after C and stuff. So the first amino acid is always methionine, which I just told you. So I might ask you that. What's the first amino acid of every protein? And it's always methionine. It doesn't matter what organism it is, except for viruses. There are initiation factors, just like we have transcription factors that have to stabilize this. In genetics, I make my students know the initiation factors, but you don't need to know that for intro biology. And that's pretty much it. So elongation is just adding change. So the artist, since we don't really, in a flat book, you can't have like a video going. They sort of made this drawing that shows you sort of a frame by frame step of what happens. So here we have the mRNA, right? It's going through the small and the large subunit of the ribosome. Remember, the large subunit has that E site, the P site, and the A site. And so the P site has this growing amino acid chain. We call it a polypeptide. It's a protein. All those mean the same thing. This codon here is going to match this anticodon, and it's going to bring in this amino acid that it's attached to from being charged. Once it does that, there's going to be a reaction. So both the P and the A sites are occupied. The reaction is a dehydration reaction to link this chain to this chain. Now we have the growing chain on the new tRNA that just came in, and this one has nothing on it. So this one is now what we call uncharged, and it's going to go back to that amino acyl tRNA synthase and get another amino acid that to become charged, and then it can come back into line. Whenever its turn comes up, then it'll come in there. So the ribosome moves, this whole thing is going to move 3 spots. So this is going to end up being the E spot site, this will be the P site, and then this will be the A site over here, which is shown in here. So this is, let's just number this A and B. So this is A, right? Now that the ribosome is moved, A is in the E site. So this is going to end up here. This is in the P site. So the B chain is in the B site. And then this C site here is open for the next tRNA to come in. And so that's what's going on. And this ribosome is going to sit there and move over 3 letters and look what the codon is. And the tRNA will come in and add the next amino acid. It'll link it to the chain, and then it keeps building this growing chain of amino acids until it hits the stop codon. Now, what I want you to know out of this is that the enzyme that does it, that does this dehydration reaction, that is called Peptidal Transference. It's transferring the growing peptide from the tRNA and the P site to the tRNA and the A site. All right, so it keeps doing that until it stops, and we call that termination. It only stops when it reaches a stop codon. And that stop codon is UAG, UAA, or UGA. And I'll show you back, we'll go back to the genetic code. And you can see these are the stop codons. So the stop codons are UAA, UAG, and UGA. You don't need to know those. I will give those to you. If you were taking genetics, I'd make you know those. But you should know the start codon is AUG. The stop codons, I don't care if you know them or not. But if you're getting a biology degree, you might as well memorize it now because you're going to have to learn it eventually. Okay, so when it hits the stop codon, in this example, the stop codon is UAG, there's no tRNA for the stop codons. Instead, there's a thing called a release factor, which has an anticodon that matches to these 3 codons. Once that release factor is in place, there's no way to transfer this growing amino acid chain to here. So when it clips this off, it can't add it to anything else. And so it just gets released. So that protein gets released, and we learned in chapter 5 that it goes off and it folds into its correct shape. And that protein can be anything. It could be hemoglobin, it could be a protein that's part of glycolysis, like that hexokinase we talked about. It could be collagen for your face. It could be, anything. So it really depends on what that protein is. I mean, it could be the proteins in milk. It could be the amylase in your saliva to break down carbs. But whatever that is, that protein is going to get released. It's going to fold into its shape, and then it's going to do its function. And whether it's a hemoglobin, it would carry oxygen. If it's amylase, then it would break down the carbs that you put in your mouth when you eat. It could be trypsin, which is in your which digest proteins, or pepsin, and both of those do that either in your mouth or in your stomach. So in the end, you've just taken DNA and converted it into RNA, and then that RNA has just been made into a primary structure of amino acids, a protein, and then that protein is going to fold into its secondary, tertiary, and quaternary shapes, like we talked about in chapter 5. And the release factor causes the ribosomal subunits to break apart. So everything at termination, when it hits this release factor, everything is released, and translation completely ends. All right. So at this point, I'm going to show you a video, and I can go to so let me share this, change the screen sharing. So if you go to the Canvas class and then you just go to pages, you guys can see this, right? I'll take that again.
Yeah, yeah.
So just go to web links, and then you can go down here, and it'll there's videos of transcription, and then RNA splicing that we talked about, and translation. So I'll show you the transcription and translation video.
DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme Rna Polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix.
And remember, those factors are called transcription factors.
The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions.
So we didn't talk about enhancers, but enhancers they bind transcription factors that may be required to start transcription. So what's cool about this is that you can see that these things can be thousands of nucleotides away on the DNA. But remember, DNA is like a shoelace, so it can bend. So you could have something thousands and thousands of nucleotides away from the promoter, where this is all assembling, and it still have an effect on transcription, whether or not transcription occurs.
Regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism.
So the copying mechanism is RNA polymerase. And so we talked about this. So this is so that first part was the initiation of transcription. And then this is now the elongation stage of transcription.
The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand.
So that each of these yellow things are representative of an RNA nucleotide, like an A, a G, a C, or a U. And remember, it's only reading one strand. So it's only reading the 3 to 5 DNA, and it's building this in a 5 to 3 direction. So the thing snaking out of the top is the pre-messenger RNA.
The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
So in the end, when it released, it hit the terminator. And remember, you got to keep, I told you guys, you got to keep these two piles separate. So transcription is completely different than translation. And so initiation, elongation, and termination for transcription is different than for translation. Okay, so I'm going to go back and we'll look at translation.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA.
So these green things are representative of the tRNA. Remember I showed you that three-dimensional shape, so they've sort of modeled this after that. And then those tiny red dots on the end, those are the amino acids. And if you forgot what amino acids are, you can go back to chapter 5 and take a look. But every one of these with a red dot on it is charged. Ones without a red dot are uncharged.
The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the Central Dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
Okay. So I'm gonna go to a different website. This is called DNA. Not for interference, but for DNA information.org, and it's a Dolan thing. And so you could go and do some interactions here. So we go to the code and you go to copying the code, So you can actually go and pretend like you're doing transcription. Maybe this is a flash thing too. Maybe you can't do it. All right, I lied. I don't think it works anymore. Oh, this one works. So this is for translation. So we have the large subunit and the small subunit. This is showing. Can you guys see the screen?
Yes.
Okay. So this is showing the codons and the anticodons. So the green triangles and then the red dots are the amino acids that they bring in. So you basically can go through this and pretend like you're a ribosome. So the ribosome is going to go, let's say that this is the codon AUG. Remember, that's the star codon. So it's always going to code for you methionine. So I'm just going to go through and find methionine. The anticodon should be U and A and C, right? Because those match. So that would be the first amino acid. And then the ribosome just moved 3 letters. So the next codon is UCA. So we just got to find the ones that match to that. So that should be A, G, and then U. And that is this one. And that would have threonine. And then it moves 3 letters over. And the next one would be A, C, G. So that would pair with U, G, C, which is threonine. And then the next one would be GCA. So GCA would pair with C, G, and U, which would be alanine. And that goes next. And then CAG would pair with G, U, C. And that's glycine. And then UAC would pair with A, U, G. Threonine, and then UAC would pair with A, U, G. No, that's a stop codon. UAA, UAG, UAC. All right. So anyway, that's I don't know why this other one doesn't work, but and basically you would pretend like you're an RNA polymerase and you would match the letters. All right. And then this is RNA splicing. Here's a video of how RNA splicing works.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear lipoproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the 2 exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes 1 intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
There's some genetics problems and some other genetics problems too here if you guys want to work on those for chapter 14. Anyway, so I'm going to stop sharing this and then we'll continue with the PowerPoint. Okay, so hopefully you understand the initiation and elongation and termination for translation. And then what you need to know is like a freeway. You don't have to wait for one car to get off before another car can get on. And so if you have a messenger RNA that's in the nucleus, and then it leaves and goes into the cytoplasm. So we would have the 5' end with the G cap on it, and then the 3' end with the poly A tail. You can have a ribosome that's going to find this in mind and then scan it until it gets to the start codon, and then that's initiation, and then it will start reading. So once that ribosome is out of the way of the start codon, another one can come on and bind to here, and so you can have multiple ribosomes on the same mRNA at the same time. And when that happens, we call that polyribosomes. It's quite common. And so that means that you can have one mRNA that makes lots of protein. And it's the same protein, right? Because only the mRNA sequence gives the amino acid sequence. But several ribosomes can translate the mRNA at the same time. And so it makes lots and lots of copies of a given protein. This occurs both in prokaryotes and eukaryotes. So it doesn't matter what you are, as long as you're not a virus, this is how it works. Viruses don't have ribosomes. All right, so once the protein is made, remember it has to fold into its correct 3D shape, and we covered this in chapter 5. We talked about primary and secondary and so on. So if you want to review that, you can. Remember, the most important thing in biology is shape. So if it doesn't have the right shape, it's not going to have the right function. If your hemoglobin doesn't get made precisely with the right amino acid order, remember we talked about that, then you could end up with sickle cell anemia, or you could end up dead. There's only two options there. So it's really important, that 3D shape. Remember that the sequence of the protein determines the shape, right? And that's the primary structure that we talked about. And then remember, during and after translation, the protein will fold into its secondary and tertiary. Remember, secondary structure is caused by the interaction of the backbone, and it's formed by hydrogen bonds. If you forgot that, you can go back and look it up in chapter 5. And then tertiary structure is caused by the interaction of those R groups that are different for every amino acid. There's 20 different R groups. So this process of going from DNA to RNA to proteins is really important biologically, and it's also really important in creating medicine as well. So there are lots of pharmaceutical companies that do nothing but look at shapes of receptors. So they're looking at shapes and they're trying to figure out how they can make a protein to fit into that shape. When they do that, right, it could be like a better insulin, because we have insulin receptors. So they're looking for something that fits better into the insulin receptor. It could be something like a pain blocker. It could be something that relieves inflammatory processes like arthritis. It could be a better painkiller. It could be something that restores your broken hemoglobin or whatever. But understanding this process is really important. And even though, you know, that these pharmaceutical companies have supercomputers that do nothing but look at A, G, C's, and T's, they translate into RNAs, and then those RNAs get made into proteins, and they look at the three-dimensional shape that they predict the proteins are going to make. And then once they do that, then they try to, do clinical trials on animals and stuff to see if it improves like insulin function and maybe diabetics or whatever. But they're not always accurate because there are other things called chaperones that help the proteins fold into their right shape. And so that's a big unknown. Like the computer models are looking at minimal energy. Right? What's the least path of resistance for this protein to fold into its right shape? But this isn't always correct because sometimes chaperones, which are other proteins, help fold these into place. And these chaperone proteins are actually called heat shock proteins because they were discovered when organisms get hot, lots of these are made. And so when you get hot, what's the danger of that could go bad with proteins? Right. And so these heat shock proteins, their job is to hold their shape as long as they can so that they still function, so that you don't die, essentially. And so that's how scientists discovered these chaperones, because they literally took fruit flies and put them out in the heat to see what proteins they made more of. And then those were called heat shock proteins. And they use those to figure out that these also help proteins, once they leave the ribosome, to fold in the correct shape. So it's not so easy to predict. And, I've done a lot of this for pharmaceutical work and stuff like that, trying to cure human diseases. And sometimes it's difficult to predict these shapes these proteins are going to take on because of these chaperones. Even after the proteins are made, they can get modified. We talked about the blood groups, the A's and the B's. That's a mannose, right? So it's actually a glyco because we added a sugar to a protein. And so these are the modifications we're talking about. They're post-translational. They have to happen after translation. So we can add sugars like the AB blood group that we talked about. And that would cause it to be either an A or a B, because if it adds a sugar in a different place, it's going to have a different shape. And those are called glycoproteins. And we could add lipids, different fats to it, fatty acid tails, and those are called lipoproteins. A good example of this is that low density lipoprotein, or the bad cholesterol that everyone's talking about. We can add a phosphate. Remember, if we add a phosphate to a protein, what does it do to it? Changes its shape, and that change in shape changes its function. We can also change the length of the amino acid chain, the protein chain. We could cut amino acids off the end. We could cut the protein in half to make more proteins, or we could add proteins together. And this is, remember we learned this is called quaternary structure. And we talked about this in chapter 5. So those are what we call post-translational modifications that can occur. So this is the very last slide of this whole class. And this is just talking about prokaryote translation is different than eukaryote translation. Remember, in eukaryotes, we have a nucleus, and the DNA is in the nucleus. And then outside of the nucleus is where the ribosomes are. But in prokaryotes, the ribosomes are right next to the DNA. So the process of transcription and translation are coupled together in prokaryotes. So here's what's going on. The RNA polymerase is this. So this is, remember, the RNA polymerase does transcription. RNA polymerase is going to find that promoter and bind to it. Remember the Tata box and all the other stuff we talked about. And then it's going to start transcribing. And remember, it can only transcribe in the 5'3" direction to make RNA. So this red line is the RNA that the RNA polymerase is making. Once that RNA is made, the ribosome can jump on that RNA. So this is translation. And in prokaryotes, translation can start before transcription has even ended. There are ribosomes on here before this polymerase has reached the end of the transcript. So prokaryotes can respond to their environment faster because their ribosomes and DNA are in the exact same place. In eukaryotes, you have to get a signal from outside of the cell that gets sent into the DNA. Then that tells the DNA to make an mRNA. Then that mRNA has to get modified and then go into the cytoplasm where it's recognized by the right ribosome, and then that's turned into a protein. So that's quite a lengthy process to go through, as opposed to a prokaryote, which just has its DNA and its ribosomes all in the same place. And so if it gets an external signal, once it starts transcription, it can also do translation and make the protein right away. And this is why we use bacteria a lot to make artificial proteins. Like, for example, there's a thing called umulin. You guys can look it up. So in humans, before we could take genes and put it into bacteria and stuff like that, people with diabetes would die, or they would have to get insulin from another source. The only other sources were humans, and most people are using their pancreas, so you can't just go steal people's pancreases. It would kill them. So you would get insulin from cadavers, or you could get it from pigs. But a lot of people got diseases from pigs because pigs are closely related to humans, as far as, like, you know, you probably heard of the swine flu and stuff like that. And so that wasn't a great source. So the first protein that was genetically engineered was for human insulin. It was done by a company called Genentech. And their stock symbol is actually DNA. And so they made this human insulin, and what they did is they took a gene out of a human for insulin, they inserted it into a bacteria, E. coli specifically, and so remember, E. coli doesn't know what DNA is in it. If it sees a gene, remember, it has to have the promoter, the coding region, and the terminator. So they had to design the gene with a promoter, and they designed that gene with a viral promoter because they wanted it to be on all the time. And then they put insulin as the coding region, and then they put a terminator in there, and they had to put the strongest terminator that science knows about, and that's from another bacteria called Agrobacterium. Because they didn't want to make an insulin that would be read past where it's supposed to stop, because it would make another protein, and then you would inject that into people and it could kill them. So the FDA required a strong terminator, and that's what they did. They built this whole thing, they put it in a bacteria, that bacteria sees this promoter, its polymerase turns this into RNA, and then once the RNA is made, its ribosomes turn it into protein, and then that protein, which is human insulin, is secreted by the bacteria and then collected by scientists and then given to people, right, to humans as a substitute insulin. And that changed the whole disease of diabetes, well, type 1 diabetes, for lots of people. And they do that for all kinds of drugs, like the drug Humira. You probably hear about it on TV all the time. That's a drug that's derived from antibodies that were created the same way. So it's, if you understand this process, you know, understanding this process is the key to developing the vaccine so fast against COVID. And it's the key to curing a lot of human diseases, or making, you know, like sickle cell anemia, or if you have hemophilia or diabetes or any of this other stuff that can be caused by a messed up protein. And the reason you have to inject this is because, remember, you have enzymes inside your stomach that digest proteins, so you have to directly put it into the bloodstream, any protein compounds. All right, so hopefully that explains how we can take DNA and turn that into RNA and turn that into proteins, which make us what we are. So I'm going to end the lecture here, unless you guys have any questions about anything.
No.
So I don't know if future class, I don't know if I'm going to make new lectures for next semester or not. I might not, but anyway, for this semester, so this is spring 2021, here's the game plan. So today is the 13th. I'm going to hold a review on the 18th, and I'll record it just like I do everything else, and I'll put it on Canvas. And then I'm going to open up the exam for everyone to take on the 19th. And then I will give you until the midnight of the 23rd. Actually, I'll open the exam at midnight on the 18th. That way you can do it right as soon as you want. And then I'll give you until the 23rd at midnight to complete your exam. You have to turn in all of your stuff by the 23rd because grades are due on the 24th. And this is weird because, remember, the semester got extended a week because of the whole hacking thing. If you want to take your exam early, you can do that. I don't care, but I don't want to open up to everyone. So send me an email, and then I can open it up this weekend if you want to just finish, if you're just ready to be done. But anyway, regardless, I have to have all of your assignments and stuff in, because grades are due on Monday the 24th, and I have to have them in by noon. So I have to have all of your assignments in by midnight on Sunday, so I can complete the grading and stuff like that. Generally, the class gets curved a few points. So if you're close, your grade's close, like you have a 77 or a 78, that's probably going to get pumped up to a B. But other than that, the grades that you see in Canvas are probably the grades that you're going to end up with. Remember, the final can count for your lowest test grade. So your final can count twice, but only if you do better on the final. So I've seen students go from having a C grade to an A grade by killing the final. So this is a really important test. So make sure you guys study for it before you take it. Anyone have any questions?
No, we have office hours next week, like Monday, Tuesday.
Yeah, so that's a great question. So next week, I actually have a department meeting on Thursday. So and they scheduled it from 2 to 4. So I can't do office hours from 2 to 3, which I normally would. But I will make them before the meeting. So I'll reschedule those office hours, but I will hold normal office hours next week. So 17th, 18th, and 19th, I'll have office hours. And then I'll also have them today, right after this ends.
Thank you.
All right. Good luck, guys. It's been a pleasure. And I will see you for the review, hopefully.
All right, thank you.
Thank you.